All right, here we go with meiosis. Meiosis, if you recall, is a reduction division. Okay, we're going to reduce the chromosome number by half and go from a diploid cell to some haploid cells. Okay, going up my little guy there. There we go. All right, so why do we do this? So again, the reduction division, go back up here. These haploid cells are our gametes, right? Our egg and our sperm. And when we fuse the gametes, it adds those two haploid cells back together to make a diploid cell. N plus N equals 2N. Okay, so if meiosis reduces the chromosome number by half into gametes, when we fuse those gametes, which is called fertilization, combines the two haploid gametes to produce a diploid zygote. Okay, so we have to reduce the number of chromosomes because we're going to add two together. So if we didn't have the chromosomes, then sexual reproduction would result in doubling the genetic material each time we went around, and that would not be good. Okay. All right. So let's move on to meiosis. Again, meiosis 1, there are two divisions, meiosis 1 and meiosis 2. We're just going to go through meiosis 1 in this screencast. It has all the same phases as mitosis, except with a 1 behind it. Okay? Prophase I, Metaphase I, Anaphase I, telophase I, and they are, of course, followed by cytokinesis. Start off during prophase I, a very busy time for the cell. And prophase I has all the regular events of prophase, but it has one very special event, prophase I of meiosis. And that is the homologous chromosomes find each other, pair up, and exchange DNA. Okay? And this is critical to creating our good friend, genetic variation. It's all about genetic variation. All right, so our homologous pairs of chromosomes pair up. Again, in mitosis, this does not happen. Proteins kind of stick them together so that the chromosome you got from mom and the chromosome you got from dad line up perfectly along their lengths. After they line up and they get all set, a process called crossing over happens. And this is where basically the chromosomes, the homologous pairs, exchange chunks of DNA. Okay? Mutual exchange of corresponding sections of the chromatids. So crossing over starts when enzymes actually cut the DNA of both of the homologs and switch the pieces, then graft the ends back together. Okay, switch the pieces. The proteins that were holding the chromosomes together then depart, but the chromosomes kind of stay stuck together where the crossing over happened, and this kind of forms a little X, and the Greek letter X is chi, and so these are called chiasmata. Okay, and this is where the crossing over occurred. We're going to see a picture in a second. It'll make much more sense. So if the pieces of DNA that were exchanged carry different alleles or different forms of the genes, then you've had a recombination, a genetic recombination. And when you're swapping pieces of chromosome, if you think about it now, a piece of the chromosome that you got from your mom is now stuck on the chromosome you got from your dad. The pieces of the chromosome you got from your dad is now stuck on the chromosome you got from your mom. So you've sort of created a whole new chromosome. You've created a chromosome that is uniquely yours. And that's the chromosome you're going to pass on to your offspring. And, then they'll be saying, oh, this is the chromosome I got from my mom or my dad. Okay, so all new combinations of genes. Okay, totally new. And this usually happens about 2 or 3 times per homologous pair during Prophase I. And that's kind of like the major event of Prophase I, and it takes a while. So Prophase I in meiosis is really long. But as I said in the beginning, in addition to recombination crossing over, all the regular prophase events happens. So again, similar to mitosis, we get spindle fibers, we get nuclear envelope breaking down, we get microtubules attaching to the chromosomes. Okay, but what's different here, actually, you know what, I'll show this and then we'll talk a little bit about prophase. No, I'm going to talk about it now. So when the microtubules grab the chromosomes, they actually grab because we have our homologous pair of chromosomes next to each other. Microtubules grab each side of the homologous pair. So this microtubule grabs this one, and this microtubule grabs this one. So when the microtubules pull towards the poles, they're actually going to separate the two chromosomes in the pair. So let's look at this diagram of crossing over. Again, we have our homologous pairs find each other, which is pretty impressive. Okay, here's my pair of homologous chromosomes. Then we get our crossing over. We get our little chiasmata. You can see the little X's that form. Okay, the DNA swaps places, and then you'll look here. The result is that, like I said, a little piece of the yellow chromosome is now on the purple chromosome, a little piece of the purple chromosome is now on the yellow chromosome, and we've created all new chromosomes. All right. Now, that's all prophase I. It's a lot. Moving on to metaphase I, again, remember the homologs line up together. We're still keeping them together because we have to make sure we get them apart. So the homologs line up in pairs at the equator of the cell. So again, the microtubules attach to one of each pair, and they're attached to opposite poles. So if you look here, okay, we have our prophase I. Again, all of our prophase stuff is happening. But I want to draw your attention here to metaphase I, where you see, again, our homologous pairs are lined up at the metaphase plate, and each half of the pair is attached to a different pole. Okay, so again, when we move on to anaphase and the pairs separate, okay, one pair is going to move towards this pole, the other half of the pair will move towards that pole. And that's how we reduce our number of chromosomes. We start here with 2n, okay, pairs of chromosomes, 2 sets of chromosomes. And down here at the end, we only have 1 copy of each chromosome. So now we have a haploid cell, and it is N. A little bit more about metaphase, though. We'll see that picture again. So metaphase, when the homologous pairs line up at the metaphase plate here, how they line up, first of all, they line up in pairs, and how they line up is actually independent of each other. So I have one pair, and then if you look it up here, the purple chromosome was on top and the yellow chromosome on the bottom. Over here, the yellow chromosome was on top, the purple chromosome was on the bottom. And how this is done is completely random. You do it again, and both purple chromosomes might line on top. Okay, it has nothing to do with each other, so it's independent. And this phenomenon actually also helps to contribute to our good friend genetic variation. Okay, and this is called independent assortment. Okay, so because it's random how the homologous pairs orient themselves on the metaphase plate, the one that goes to each pole is going to be random. And this means that the daughter cell can receive any combination of maternal and paternal homologs. Okay. And again, this random combination is called independent assortment. Okay. So on the metaphase plate, the homologous pairs assort independently of each other. Okay. So recombination during prophase I and independent assortment on the metaphase plate are responsible for, again, for our genetic variation, for genetic diversity of the haploid cells produced by meiosis. And, again, we can talk in class about the infinite number of combinations we can get from those two processes working together. All right, so let's move on to anaphase. We've got all of our homologous pairs lined up on the metaphase plate. Anaphase, the homologous chromosomes separate. We separate the pairs. It's like taking a left shoe and throwing it in one room and a right shoe and throwing it in the other room. Sad, but it has to happen. Okay, so in anaphase of mitosis, sister chromatids separate, but here they stay together. In anaphase of meiosis, the sister chromatids remain attached and they move as a homologous unit. Okay, so we'll talk about telophase I and then we'll again look back at the pictures. Telophase I, now we have two haploid clusters of the chromosomes at either end. And they do kind of normal telophase stuff. They spindle fibers disappear, the chromosomes decondense a little bit, but they're going to immediately recondense. There's cytokinesis. Nuclear envelopes may reform, but they may not, because remember, we're going to divide again in meiosis II. Okay, and telophase is going to usually immediately followed by meiosis II. There's no intervening interphase in between them. Okay, and another good point, chromosomes do not replicate again between meiosis I and meiosis II, and that's how we keep our haploid cells. So let's look at that picture one more time. Okay, so prophase I, homologous chromosomes find each other, get recombination and crossing over, and then they attach in pairs to separate poles. Okay, how they line up at the metaphase plate is random. It's independent of each other. Anaphase I separates our homologous chromosomes. One travels to each pole. If you notice, they're still duplicated. They still have two sister chromatids each. And then telophase I, we get two sets of haploid nuclei forming. Over here, we had diploid nuclei, 2N, 2 copies of each chromosome. And over here, we have N haploid, 1 copy of each chromosome. And we have reduced the number of chromosomes, thus endeth our reduction division. Okay, stay tuned, because meiosis II is up next.